OxLamp technology used by GeneSC novel coronavirus 2019 nCoV nucleic acid detection kit is developed at the University of Oxford and distributed across the Chinese mainland by Shanghai GeneSC Biotech Company Limited under exclusive license. Rapid novel coronavirus detection should be carried out in two independent spaces. After the sample is taken in the sampling area, it should be sent to the detection area by specially assigned person for detection, avoiding contamination. The staff should take the safety precautions, including wearing masks, protective clothing and gloves. And cleaning hands with alcohol. In the sampling area, the staff should clean the bench with 10% 84 disinfectant. Take out the swab and a virus preservation solution. And insert the swab into the nostril of the person to be detected in the perpendicular direction to the nose until feeling there is obstruction. Keep the swab in the nose for 15 to 30 seconds. Then gently rotate it for 3 times. Pull out the swab and put it into the sample preservation solution. Break off the swab rod and place the swab completely in the tube. Tighten the tube cap and make a mark. The staff in the sampling area should put the samples in the delivery area. Which will be delivered to the detection area by a specially assigned person. The staff in the detection area should clean the bench with 10% 84 disinfectant. There are 2 ways to process the samples. One is to extract nucleic acids, and the other is to directly load the sample. The nucleic acid can be extracted by using an automatic nucleic acid extractor, GeneAct NL48. And a supporting nucleic acid extraction reagent GY1. When directly loading the sample, preheat an inactivation device to 95 degrees centigrade. Shake and mix the swab preservation solution for several times. Place in the inactivation device and heat the solution at 95 degrees centigrade for 10 minutes. Take out the heated solution after 10 minutes and cool it to room temperature. Preheat a heating block with the hot lid to 65 degrees centigrade. Buy a metal bath or a PCR amplifier or a lamp real-time monitoring system independently developed by GeneSC. Transfer an 8-tube stripe and cut it according to actual needs. For example, if there are 4 samples, cut off 6 reagent tubes, 4 for samples and 2 for negative and positive quality controls. Open the tube caps from the side edge to prevent contamination. Adjust a pipette to 25 microliters. Press the pipette down to the first level. Transfer 25 microliters of the purified nucleic acid sample or the original heat-treated sample to a PCR reaction tube containing a lyophilized reagent. Then press the pipette down to the second level and discard the pipette tip. Transfer 25 microliters of the negative quality control to a new PCR reaction tube as the negative control, and transfer 25 microliters of the positive quality control to another new PCR reaction tube as the positive control. Seal the 8-tube stripe with caps after sample loading. Shake or flick the 8-tube strip with fingers, and centrifuge the tube briefly to place the solution on the bottom of the PCR tube. The color of the solution should be red. If it is not red, please replace with the new PCR reaction tube. Put the PCR reaction tube in the preheated heating block, 65 degrees centigrade immediately. Take out it after reaction for 30 minutes. And check for color changes after cooling to room temperature. If the color remains red, the result is negative. If the color turns yellow, the result is positive.